Your brain is an amazing thing. It helps you in everyday, really mundane activities, but it would be impossible without short-term plasticity. You wouldn't be able to start a sentence and then remember how it started by the time you reach the end, ironic, and also be able to maybe have a telephone conversation, but also write or watch a television show. Keep those two tracks separate and understand them. It's a crazy thing. And it relies on our friend Cara to tell us more about it in a proper way, because I'm going to be a layman.
So there's an interesting study that came out of the Max Planck Institutes of Experimental Medicine and Biophysical Chemistry that is really getting down to the heart of exactly how this plasticity works. So we've known for a really long time that cells communicate both chemically and, what's the word I'm looking for? Electrically.
So synapses and neurotransmitters.
Exactly. So when you've got two neurons in your brain, they're going to communicate with one another across a little space called the synapse. That's where they connect. And there's both an electrical phenomenon, which means that the neuron before is going to fire. There's also a chemical phenomenon, which means that it's going to release little packets of neurotransmitter. Okay. You know, whether it's serotonin, dopamine, norepinephrine, all these things we've heard of. Now, we know that the way that that neurotransmitter is delivered is that it's packaged in these little things called vesicles. It's just a little part of the cell that's membrane bound, that's full of neurotransmitter. It moves to the membrane where the synapse is, and it releases those vesicles. And it seems like a lot of this kind of neuronal flexibility, this plasticity, this learning, is dependent upon having enough of those vesicles ready to go, right? There's only so much neurotransmitter that's packaged in these vesicles. There's only so much that they can give off. These researchers have shown in vitro previously, which means outside of the animal, in petri dishes, playing with cells, that this vesicle binding is dependent on the calcium-calmodulin complex, which is an enzyme. It's a type of protein that's involved in neuronal signaling. It tells these vesicles to be ready to go to release whatever neurotransmitter they need to release. And they're showing that the number of immediately releasable vesicles at the synapse determines how reliable that synapse is. So if there's more vesicles full of neurotransmitter ready to go, that synapse is going to be more highly functional.
So they found a way to possibly make that happen, to supercharge the nerve cells.
Well, that's the interesting thing about reading science in the press, is that no, they have no idea how to supercharge them, but they know what does supercharge them.
It's funny that it was on the title, and then I read it, and I was like, wait, where?
Where, exactly. And that's the thing. Because now they know why nerve cells are sometimes more effective than others, because it seems to be almost dose-dependent on the amount of available vesicles full of neurotransmitter, which seems to be dependent on how much calcium-calmodulin is present. They now feel like, okay, this is not just a phenomenon that we've seen in vitro. It's a phenomenon that's happening in the human all the time and at every single neuronal connection, at every single synaptic connection. All the time,
constantly.
All the time, constantly. So there's a great quote at the end where the researcher says, "We have discovered a key molecular mechanism of short-term plasticity that plays a role in all synapses in the brain, and not only in cultivated neurons, as many colleagues believed." So, I mean, they're coming out there and saying it. Every single neuron in the brain relies on this phenomenon. They rely on calcium-calmodulin, which is a regulating protein. It's a signaling protein in the brain, an enzyme that tells these vesicles, fill up with neurotransmitter, move your way to the membrane, dock there, and release neurotransmitter into the synapse so that the next cell can take it in. The more calcium-calmodulin, the more available vesicles, the better the synaptic plasticity. The better you will learn, the more highly efficient your neuron cells will be.
I like the sound of that. How do we produce more of that enzyme, though? Is it possible?
I mean, that's an interesting question. It probably is possible. You know, it all comes down to chemistry. I'm not sure if there's a way to take this enzyme exogenously, or if that could wreak total havoc on the brain.
I'm a little bit worried I'll ruin my brain
exactly.
if I'm taking whatever produces this enzyme.
You know, the interesting thing is that a lot of people think that like more is always better, but we know in neuroscience that you start out with way more neuronal connections than you end up with as an adult. You have all these connections at the time of infancy, and then they start to prune as you get older, and those more specific connections are strengthened throughout environmental experience, and then you have an actual functional brain. If everything's connected to everything else, it'd be really hard to get anything accomplished. And a lot of people have, you know, you hear about that fallacy of like, people only use 10% of the brain, which...
I'd heard that's like
it's total bullshit. It's total bullshit. You maybe, I mean, who knows how much of the brain you use at any given time, but of course you use your entire brain.
Yeah.
But the truth be told, if all of your cells were firing at the same time and releasing neurotransmitter, that's what we call a seizure. That's really dangerous. You don't want excessive brain activity. You want very specific, focused brain activity.
We forget that the brain does other things besides work out problems.
Yeah, the brain does everything. It pretty much is who you are. It does everything. Pretty important.
So be careful of that.
Yeah.